Good morning, everybody. I think this is record attendance for a Sunday morning, actually. So thank you all for getting up and being here. The basketball game didn't go too late, I don't think, and now we don't have to watch any more basketball for another year. So that's, I guess, or hear about it, if you're. Welcome to Sunday morning session. Thank you all again for being here. Thanks for a great meeting. For all of our sponsors making it happen, all of our speakers, and then also everybody who's spent the weekend with us. We've got our last industry sponsor symposium this morning, and it is for 0.75 Category 1 credits. So it is in compliance with CME. We've got our pre-test QR code, just like we've done in the other session. So if you guys want to scan that, just so we can get a baseline, and then when you do your post-test to claim your CME, we can compare. So I'll give you a minute there.
I think we can.
And then we'll. All right. I'm gonna. Whoops. Oh, sorry. Everybody's still. Sorry, guys. So this talk is going to kind of, I think, really nicely summarize and hammer home some of the stuff we've been talking about all weekend, and it's going to give us a chance to kind of consolidate, look forward to the future, and so a little bit of, I don't want to say repetitiveness, but we're going to be revisiting some of the exciting stuff from the last year, all these updates. And then kind of talking about some of the future directions as well.
Open at your place?
That's me. Honored to have our society president, Dr. Endometrial, and Dr. Martino with us to be faculty for this. And here are our disclosures for, like has been said before, not just us, but everybody that reviewed our slides for CME purposes. So we're going to talk about ADCs. We're going to talk about clinical data, key and emerging. We're going to talk about kind of up and coming targets, and how to sequence. We're going to talk about a little bit about sequencing, and then suitable clinical trial opportunities going forward. I think I already used my 3 minutes, so I'm going to turn it over to Dr. Endometrial. This is going to be quick, and once again, we're even better set up here for a conversation. So we'll definitely be hoping for some participation, which I think the first ISS, nobody raised their hands when I asked people to raise their hands, and then by the end, everybody was arguing with the people that were standing up here. So let's keep that going. Let's actually we'll actually have some good discussions about the cases, and then we'll move on. So thanks again to GSK for sponsoring this session with an independent medical education grant, and Megan.
Good morning. I'm just going to start with the basic stuff that we've actually have heard more and more about. So my talk's pretty easy because you all know this already. Oops, wrong way. So I'm not going to go over this slide because actually Marty is really going to go over it. But as you recall, ADCs, this is basically the basic concept of them. And the most important part of it, I'll have Marty go over that. But why are ADCs in gynecologic cancer? Why do we want them? They have high expression of targetable antigens. We need more effective therapies in recurrent cancer. We all know that. And potential to improve the efficacy while really limiting the toxicity, and then expanding the biomarker-driven treatment options. So these are why we wanted to look at this. So and we're going to be just concentrating on ovarian and endometrial cancer, and the two FDA-approved ADCs are MERV and TDXTD. Both of them have been approved by the FDA. looking at the targets of the folate receptor and HER2. And in ovarian cancer, it's the folate receptor positive, which was FDA approved in 2024 for second line plus. And in endometrial cancer, as well as ovarian cancer, and also our cervical cancer that commonly gets left out, it's HER2 positive tumors. And this was accelerated approval, so we're waiting for final data to improve it. And so we're going to just go right into the MIRV, which is targeting the folate receptor alpha expression in epithelial ovarian cancers. And 35 to 40% of platinum-resistant high-grade ovarian cancers express this high level. So the Marisol study was the one that helped get this approved, and as you know, it's an open-label phase 3 randomized controlled trial of MIRV against investigator choice, looking at the stated endpoints. And as you could see, as Jubilee had pointed out, you could actually put your thumb in between the graphs if I had it correctly up there. There's good separation in both progression-free survival as well as overall survival. And if you look, it improved the overall survival. You know, not huge because we're dealing with platinum-resistant disease that's had multiple treatments, but significantly better than other treatments have previously shown. And so this has been an amazing improvement in our platinum-resistant patients. Now, with regards to safety summary, like overall compared to chemotherapy, it's but I call it about the same. I think patients do tolerate slightly better dependent on the patient when it comes down to it, but the data does show that it does have a tolerable safety profile. And when you actually take a look at the slides of the things that really matter, in the majority of the patients, the hematological side effects are much improved, which in my practice, a lot of the patients have already had chemo, and so their bone marrow is very touchy with giving more treatment. So this is really nice for the patients because they don't have to get Neulasta or Neupogen as much, which actually gives them a lot of the side effects that they really don't like with regards to it. Peripheral neuropathy, slightly less, however, can be pretty significant with MIRV if you've had patients on it, which can be a limiting factor, especially if they've already had neuropathy. I recently just had a patient on it who did not have any neuropathy with her prior chemos, with Taxol, being treated with Taxol multiple times, and then was placed on it and had horrible neuropathy that we had to end it, even though that she had a great response to the treatment itself. And then with regards to the adverse events, ocular side effects you will definitely see. In private practice, this was like where I had a lot of issues with the ADCs, trying to figure out how I was going to get my patients to get eye exams and everything. Because I don't have the full university base or hospital practice, but we actually in the community have had like great responses to all the eye doctors with regards to it. So this actually has not been an issue whatsoever. So that is always a good thing. Now, since this was FDA approved, it has been placed in the NCCN guidelines in recurrent platinum-resistant disease. And as you can see, it's on a preferred regimen for these patients. And if you remember, the folate receptor alpha expression should be 75% or greater. However, it also is on the NCCN guidelines in combination with bevacizumab if your patient is greater than 25%. So this can be used in those certain circumstances. In addition, in platinum-sensitive disease, it also is in the NCCN guidelines for both combinations as well. Now moving on to targeting HER2 in ovarian and endometrial cancer. Again, we have T-DXd. I'll have Marty go over that again. With regards to the background of HER2 and gynecologic cancers, I thought this was a great slide showing us, comparative ovarian, cervical, and endometrial cancer, what percentage of the patients are HER2 positive. And as you can see, ovarian cancer is actually pretty small, which is in a sense, you're kind of a little bit surprised because you think of, patients with BRCA, but when you actually look at HER2, it makes sense, but why does breast cancer, have it, So I had to actually, really look into the science of that. Now I understand. And then when you look at cervical, you're like, well, why would HER2 be positive in cervical cancer? But if you actually look at, the data, it's mainly the adenocarcinomas of cervical cancer that's positive for HER2. And then when you look at endometrial cancers, when you try to look at, okay, what subset of endometrial cancers are HER2 positive, uterine serous carcinoma can be up to 40% HER2 positive, which is really interesting to know because maybe this is going to be the great thing for serous carcinomas of the uterus, but not necessarily serous carcinomas of the ovaries. So it's interesting data. Destiny trial, obviously preliminary data, so they can't break that out, nor have they, with regards to that. Now, so as we know, this is from the phase 2 Destiny Pan-Tumor 2 trial, where they basically gave T-DXd to a pan of tumors, including endometrial, cervical, and ovarian cancer. And just of note, they used the gastric HER2 scoring method, not FISH. Which most of my pathologists do that currently. And when you look at the responses, breaking it down, endometrial had the most responses with cervical being second. So we have about 84.6% with HER2 3 positive, and then still 47% with HER2 2 positive. And then cervical cancer was 75%, which I thought was amazing. Now, if we look at our plots, you can see that there is a significant difference in endometrial cancer, as not such a good response in ovarian cancer overall. And this is both with HER2 as well, HER2 3 positive plus HER2 2 positive, and so therefore it was approved for both, pathological findings. Side effects, this is actually fairly well tolerated. You're going to have some nausea on the drug, but this is very well controlled with our medications that we have. You do see a little bit of anemia, and fatigue can be quite a big concern for the patients because we've already treated them with a lot of cancers already since this has been approved, and this is probably the biggest side effect that my patients actually have is the fatigue, and I think it's just built on from the prior treatments that they have. They never recover from it overall, but patients tolerate it really well. Now, when we look at the more serious side effects, the risks are pretty much the same as all of our other drugs with pneumonitis. One of the things that I'm always interested in is the cardiotoxicity, because you're like, oh, you have to get echoes or, routinely with these drugs and everything. And overall, the risks of cardiotoxicity are very, very small when it comes down to it. And so, can we not have to do an echo so often and stuff like that down in the future? Hopefully, maybe not. The patients are, my patients are sick of getting them routinely. And I think that is what kind of all led to the new NCCN guidelines, adding this for not just ovarian and endometrial, but also cervical cancer, giving us even more options for our patients based on tumor markers. And I think that's it for me.
Thanks, Megan. Good morning, everyone. The world's changing, as we can see with these ADCs, and there's only a few of these that are here now, and as Brian will tell us and others, there's hundreds, as we know, that are coming. The best way to think about how to package this up, as I was thinking about this antibody-drug conjugate, it really gets us to precision medicine and that personalized medicine. So we put this into 3 buckets and 3 pillars, and each pillar will help shape the efficacy, the toxicity, and resistance. So if we look at the targets are like the addresses of our homes. And these are 6 of the targets that we're going to talk about: FR alpha, HER2 and 3, TROP2, B7, H4, and NaPi2 and CDH6. The payloads and the linkers will help deliver that package. So the targets or the antibodies, that gets us to the address, and then what gets into the houses or into the cells are the payloads and the linkers. Now the payloads are that drug, as you can see here, and the linkers provide that stability, cleavage, therapeutic index with that. So there's no single target is universal. We have to select this by histology. There's the highly selective targets, as we've seen, the FR alpha and NaPi2b. They are the broad expression of targets: TROP2, HER2, B7, H4, and they're across multiple histologies. And then these emerging targets, CDH6 and HER3, and some data in clear cell and endometrials as well. Here's an image, as you can see, to get into the expression profiles for some of these. They're FR alpha is the most tumor selective, and you can see that over on the left side. You can put your HER2, the HER3, CDH6 as emerging options, and then the TROP2, B7-4 have a broad spectrum of coverage with that. So what does the data tell us on these targets now? So we're looking at the personalized targets. The alpha is highly selective. It's validated with scores. And the limitation is there's some drops in platinum-resistant disease. The TROP2 tells us the advantages are strong in cervical and endometrial. Limits are lung GI expression, raises the toxicity risk. HER2 and 3, broad actionable, as we've seen over the past several decades, limits overlapping resistance. And then some of these newer targets, the advantages of B7H4 and the others, high ovarian endometrial expression, and that's important. Limited target overlap, and these are being developed currently. So for payloads, so those are the targets and the antibodies to the address. Now the payloads are the warhead, right? They define the weapon and how this is going to be delivered. There's potency to look at, cell kill mechanism, tolerability, and the bystander effect. So we look at these 4 payloads and some distinctions. The first 2 on the top, we've got microtubule inhibitors. They're highly potent, but there's a bystander effect that are with those, as we see in the side effects. The bottom 2 are topo 1 inhibitors, membrane permeable, and some bystander effect as well. And this is just a few of the payloads that are in development. Now, the membrane's permeability will change, right? You have the address, the package is there, who's knocking on the door, who's opening the door, how does that package get in? And that's why it's important for that cell to be permeable or not. So selecting that target's important, and then once it's delivered, there's a bystander effect. And that's how there's neighbors of these cells that can also be affected. Looking at the linker biology, there's stability, cleavage, and therapeutic index. Is that package going to be delivered? It's in your house, but it's now going to be delivered through the door. So there's cleavable linkers, there's non-cleavable linkers, and then there's some that are stability versus the effectiveness of these. If they're cleavable, sure, the package is going to be delivered, it's going to be released, enable the bystander effect. And that's oftentimes what you're looking to achieve. If they're non-cleavable, then they're stable. It'll hold on to it more. It requires a little bit more for release. And there's that balance that we have to determine between toxicity and cell kill. Now, how to manage the toxicities? We know there's the ocular toxicities, looking for conjunctivitis and keratitis, and I will say the ophthalmologists have been superb in our communities. It's great, just to befriend them as we all do. And if not, talk to your ophthalmologist and say, hey, I've got this new practice, new program we're doing, will you help? And they often do. ILD pneumonitis as well, that we see. The neuropathy, may require and will dose reductions and holds, maybe gabapentin if needed. And then hematologic toxicities as well. So putting it all together from a synthesis, we've got Merv looking at the FR alpha. We've heard a lot of great information about that, and we've seen overall survival benefit. Trastuzumab for HER2, we see that as well with HER2 activity. We've got tisotumab. We see that for cervical cancer, and we've got these TROP2s as well that we'll be discussing. Targets, key ADC trials, just looking at some, we've got the Mirasol trial that we now has approved, Destiny with positive data, Innova TV is approved in cervical, we've got Ascend and TROP-Fuse and GOG3095 for endometrial cancer, phase 3, and emerging data over here for the NaPi-B2 and B7H4 trials. So moving to the future, we're definitely pushing the boundaries, and this is clearly going to be the future for all of us to have to understand, and these will evolve and change. And hopefully, the side effects as that as the payload and the linkers change, there'll be much less side effects. Hopefully, getting rid of the ocular toxicities or decreasing them, and that's where the bispecific ADCs hopefully will come into play. There's ADCs and immunotherapy that we have to look at. ADCs and PARP inhibitors, higher constructs for better drug delivery as well, and potency of these, and just newer payloads as well with Topo2 and RNA polymerase inhibitors. So key takeaways, targets are not all equal. The address is not all the same, but you need to have that, the match histology to the biomarker, which is why all of us are now, remember back when we started to order different testing, it was very questionable. Some of our medical oncologists were like, well, I'm not sure we really want to have a Caris or one of these tests. Now it's like, hey, where's the Caris? Can we have that? So we really did help lead the way. And same thing on cell-free DNA. Rob, credits to you for presenting here 4 years ago, maybe, your first early studies, when no one in the not many people were doing that at all. And I'd say Florida really led the way on the cell-free DNA, thanks to this FSGO and taking the risk to go out there. Payloads drive toxicity, which is another takeaway. Linker chemistry is going to be important, how they're cleaved and when they're cleaved and if they're cleaved. And then resistance as well is multimodal. Thank you.
Before we get to discussion, I just wanted to point out, Marty did some unbelievable microscopy here to get these pictures. And then he even put in a picture of himself getting the shots. Great work in the basic science lab. So
he was bragging about it, like when we were going over the slides.
So I want to talk about a couple cases. We'll kind of start a little bit on the basic side, and then we'll move forward. So this is we all have this patient, 65-year-old with a somatic BRCA1 mutation. She got upfront treatment in 2023. She saw a PAOLA-1 regimen, was on olaparib for 20 months, and right as she was nearing the end of her PAOLA-1, she had widespread carcinomatosis. At that point, I treated her with carbo-PLD and Bev, and unfortunately she progressed after 4 cycles. She had a 5-centimeter mesenteric mass. We biopsied that, and this is fresh results from that biopsy. This isn't the original tumor. So folate receptor 1, 40%, HER2 2+, and PD-L1 positive. No trials available at that time that she qualified. She didn't want to transfer out. So what are we thinking for treatment options for this patient, with everything from the whole meeting, not just from this morning, but what are we excited about for her? She's great. She's a performance status of 0, and the only thing is that she's frustrated that she's failed, that she's now platinum refractory, and so she's very, very nervous. But otherwise, you wouldn't even know she has disease. Not bad neuropathy. 96. Yeah, absolutely. Yeah. A lot of yeah. Still got a couple of years ago we didn't, right? How does you've got you have Rosella here also. She's HER2 plus, so you have TDXD on compendium, not on label. Folate receptor. You've got MERV with Bev based on NCCN. So what are your sort of, I mean, you guys jump in too also, but what are your, how do you pick between these things for a patient like this that's otherwise a total blank slate and you don't have anything pushing you in one direction or another, all other things being equal? What's your go-to in this situation for a platinum refractory patient?
I'll say I first asked, like, okay, what are her goals? I have the big conversation of like, okay, as you know, we're not going to cure this. Quality of life is extremely important, but treating you is important to keep you, alive, but in a manner that you're not going to have a lot of side effects. So the neuropathy is one thing because, the Taxol and Prem is, a good option for her. Also, a lot of times the patients don't want to get chemo weekly. So that is a big brick wall to some patients. Like when you tell them what the protocol is, they're like, I have to do weekly, I want to do this, I want to get to that and do this. So sometimes what the patient desires in their treatment regimen will guide me on what the next step is, but I will offer them all that's available so that we could have that conversation and make that decision together.
Do you find that like kind of muddies the waters even more? I feel like if I give more than 2 options, and we're talking the difference between B96 and Rosella. Those are all right. This is both weekly Taxol regimens. Let me talk to you about cortisol receptor. Let me talk to you about immunotherapy, and then we've also got these targeted agents. Is it like?
Yes, it muddles the water. However, I also have this big institution in my area, and the patients commonly go for a second opinion, and they'll bring up a whole different treatment that I'm just like, really? But if I hadn't mentioned that, the patients will just boom, go away and never see me again, because the bigger Cancer Institute told them that that's the regimen that they had on, and I never ever brought it up. And there may have been a reason that I might not have brought it up, because I don't think they're going to tolerate it very well, or I know the data. Even though I'm a private practitioner, I know the data. I keep up on it, and stuff. But it's quite amazing. So I've learned that I just kind of mention all of them, but I will tell them these are the 2 regimens I think you should be on because of this, and this, for those reasons. So that's where I've learned that muddling the water sometimes is a better thing in my area when it comes down to it.
Yeah, I mean, when you translate it into Creole, it gets even tougher.
Yes.
In Fort Lauderdale.
I don't have that problem.
So, what you were going to say something, Jason?
How old is she?
I don't remember. 65. 65 years young. Yeah.
So she's still relatively young. She feels good, and she wants to maintain her quality of life. And I would focus on toxicity concerns first. She's got a whole ton of options. She's platinum resistant. She's likely going to probably receive multiple, several of these regimens at some point throughout her remaining life. So I would give her the quality of life that she needs at that point. Really probably the least toxic option, and probably the one that's going to give her. Then you have to think about, with B96, you got weekly Taxol, so that's kind of, from a treatment standpoint, kind of labor intensive. So those are all things that you got to factor in.
Yeah, I mean, I think we were talking about, I think it was at lunch on Friday, kind of thinking about targeted agents first, and then going into and then going into the non-biomarker specific. I think that's where I go, but I don't know. What do you think, Brian?
First of all, Megan, I know you know what you're doing, so I've seen some of your patients. So don't let the big bully down the street tell you different. I don't. The biggest question industry has, and I call them that all the time, is, Jason, which one are you going to use? Why? And which one are you going to? It's like they call me up, and I like working with them. Of course, people would say, which one are you going to use? Why? Like 3 weeks after the FDA indication. That's not the question. The question is, what order are we going to use it?
Yeah.
Right. So I think looking at it that way is different. For example, this patient has 4 regimens available. 2 of them are weekly taxane backbone regimens. 2 of them are Bev backbone regimens. So whatever order we give, because you're going to have to give the Merv with the Bev, and you have to give the P96 with the Bev. Whatever order we give, I just want to prevent doing those back to back. Right. So here maybe I'd start with Merv Bev, and then you can go with Rosella, and then you can go with trastuzumab deruxtecan, and then B96 or something like that.
Is there for sequencing? Well, yeah. I mean, I think that's unanswerable. But is there a concern for you with Merv being microtubulin and then going back to the taxane, or is that too much in the weeds?
I mean, yes, but it's too much in the weeds.
Yeah.
There's so much cross-trial things we do in our head when we're figuring out sequencing. As long as you're not eliminating anything, but in this case, if it was folate receptor 75% or higher, I would probably definitely go with Merv first.
Yeah.
Probably definitely. If she was HER2 3+ or higher, I'd go for that sooner than first. The B96 Rosella is a toss-up. Which one you use first?
I mean, that's why I kind of leveled this out, because I didn't want there to be an obvious. I figured with 4 possible good options, and then there's also a really, there's a really, I guess, but with 4 good options, I thought we'd get 5 or 6 answers. Yeah.
I also feel like I want to make sure I get to use all of them. If you don't think you can get 2 regimens of weekly taxane in, then I'd go with Rosella, because you can bounce back to Pembro Cytoxin. If you don't think you get another play. And I don't want the Merck people to hate me for that, but I mean, that's just the reality is you can give it different lines to Pembro. You can't, there's only one shot on goal with the Rosella.
Okay, so Rob, you mentioned yesterday using Cytoxan with Pembro, and obviously that's not on label like B96 is, but I always thought, Brian, you and I talked about this early on, like a year ago, like, should we be thinking about that regimen with Cytoxan Bev Pembro if we're gonna be thinking about using Rosella, so that we're not going back to back with taxane, or are we muddying that data because that's such a small trial, and we're going against phase 3 with phase 2?
I really use it with taxane tolerant. My philosophy is always go over your best. I think the data on B96 is outstanding, so I would prefer that. But I'm saying if you have patients who are still neurotoxic,
Yeah.
Or they just came off of the
So this, obviously, we've talked a lot about Rosella this week. Excellent data in Rosella, excellent data in B96. I put these here just in case we wanted to talk about it. So we put her on, again, no wrong answers, but we put her on NABPAC with relacorilant. She progressed after 6 cycles, and guess what? Now she does actually want to go to see Brian at Mount Sinai because I don't have any trials open for some reason. I don't know. But she's willing to transfer out for trial. At this point, obviously, I think we favor clinical trial if we have. What are you most excited about in the trial space for this patient here? Like what kind of holds the most promise for her going forward? What?
Yeah, I think that it's a great question. I mean, and truthfully, if you're talking to the patient, you said, "Oh, let's try one more, then go to a place for a trial." You want to reserve what you've done, the opportunity to go on a trial for a good topo 1 ADC trial.
It's coming. It's here. Yep.
That was pretty game changing. When we saw SGO was 62%. But you can't do cross-trial comparisons. You can't. You could say it looks active
Yeah.
and is worthy of investigation and looking at toxicity profile.
Yeah, I'm super. I mean, like we talked about, I think there were a lot of exciting. We talked about this earlier. That there were a lot of exciting presentations at SGO of drugs that we are all in the process of opening trials for, so that we can say who in the state has the Lilly trial open, who's going to be opening. B7H4 is going to have 2 protocols in endometrial and 3 in ovarian. This is the ovarian, one of the ovarian protocols, ovarian 01, which is in recurrent in PROC, right? Yeah, PROC, yeah. And this is against physician choice chemotherapy. I'm very excited about this. I'm very excited about RDXD. I fought really hard to try and get this trial open, and we didn't get picked for this, and I was bummed. But Brian, you had this open, and I think had this open too, right?
Listen, don't let Scott's frustrations get you down. If you guys want to do research, and I know you're out doing, you need more protocols, reach out to us. Reach out to Jenna, who's here. Reach out to me. We'll be able to figure out a way to help you out as best as we can. But I had the Rejoice 01 open, and that's open to accrual. But one of the things that we mentioned briefly yesterday, but we highlight here today, a lot of the control arms of these studies are already antiquated, right? For example, the 3096 study, I'm not just calling this one out, but it's others. While we were developing the trials and developing the science, we got the 2 FDA approvals, and suddenly I'm required to tell my patient if they want to go on to a Rejoice 01, which is new ADC versus weekly Taxol Bev. I have to say, well, give the control arm actually maybe suboptimal because of B96 and because of Brazil. We haven't changed the control arms yet, but that's one of the problems with constantly getting more approvals. We have to constantly change what the standard of care is.
So let me ask you guys this, because this is something that I'm struggling with right now as I'm about to open Lilly. How many places your site has one of these PROC trials open with the Aurelia like monotherapy, Taxol, PLD, Topo, single agent? That's like one of these antiquated control arms now. How many of you have one of those open or about to open them? Yeah, we do. And what do you do now? Which of those do you pick? Because I know I want to pick weekly Taxol, but is that going to burn me for Rosella or B96 later on? And then am I giving Doxil and I know it's not as good as a single agent?
Yeah, there's 3 options. One, not to give the trial, to give Rosella B96 to start.
Mm-hmm.
Some trials are going to increase the number of line therapies to allow for that. The second is the trial is going to have to change it if it's a new trial that's opening up. They'll probably have to change the control arm, right? And then the third issue in the trials that have completed accrual that had Aurelia as a backbone, it's going to be a review issue if the data is good enough to meet the current standards.
Yeah, that's a tough situation because 100% clinical benefit rate is amazing. So, all right, really quickly because we're running out of time here. So, for endometrial, this is a patient that this is also a real patient of mine, and I did have this conversation in Creel. She's a 55-year-old with 4B uterine carcinosarcoma, upfront surgical debulking with no residual disease. And she was pMMR, HER2 2+, FISH negative, P53 mutated, ERPR negative. So I put her on 6 cycles of first-line carbopaclitaxel dostarlimab. We continued dostarlimab maintenance for 11 months. She did fantastic. She tolerated it really well. And then all of a sudden, out of nowhere, and we see our patients before every cycle of even maintenance therapy, so it really was out of nowhere. She shows up to the ER with partial small bowel obstruction due to carcinomatosis and loculated ascites. So here are my biomarkers again. Who's going back to chemo in this setting? Anybody? who's going to Linpim here? She just came off maintenance. She progressed on maintenance to starlimab. Anybody going to Linpim? Thank you, Vagan, because that's what I did. You guys? Yeah. Oh, thanks.
would.
Yeah.
I mean,
And what else? Rerun the HER2. Obviously, so we do our upfront with breast scoring because we have GY026 open. So I want to know if I can put her on Herceptin or Perjeta in an upfront. Of course, since it's an NRG study, we don't have a translated consent. But I want to know. So I want to rerun this and see if HER2 was an option for her. But I ended up putting her on Linpim, which I hadn't done a lot of yet because it hadn't happened yet. You guys have experience with that? Does it work?
yeah.
After dostarlimab? Yeah, I was like very pleasantly surprised. I don't know. You have other thoughts about standard of care?
Yeah, a couple options. And I agree. I mean, everyone in the world is saying what you guys are saying. Yeah, it works. Yeah, it works. The reality is, when I look at retrospective data, it probably works just as well as Adriamycin or weekly taxane. So it probably works just as good as the best thing that's out there. So I'm not yet a big fan of IO for IO, especially in the PMMR patient population. There's some data like nipi-evo combining things more. I'm not a big fan yet, but people are using it a lot, and it seems to work, so you can't go against it. The only correction I'll have for you, Scott, is I just learned this like last week. Carcinosarcomas don't require HER2 staining for trastuzumab deruxtecan. Based on like the status trial, statis or something trial, there's data of carcinosarcs with HER2 zero that you NCCN listing to get trastuzumab deruxtecan.
That's awesome.
I didn't realize that.
That's great data. Yeah. So how about clinical trials for endometrial cancer? I think it's kind of the same set from everything we talked about this weekend. Obviously toxicity and sequencing, but this is TDXD. Megan did a great job of breaking that down. And then once again, MoRes. I'm very excited about this, and we're going to be opening this in recurrent endometrial. This is 1 to 3 prior lines in endometrial, prior platinum and PD-1 required, and prior B7H4 and Topo1 payload excluded. And I know we haven't really touched on that all weekend, but now that we've got all these upfront topoisomerase payload trials, it's hurting me as I'm trying in later lines trying to accrue patients because they've already seen. Maybe she's already seen TDXD. But once again, I mean, this is obviously very early days, but super exciting data. So I'm very excited about MoRes and endometrial. This is upfront TDXD in endometrial, and unless they had gotten adjuvant or neoadjuvant, so they can have gotten. It's upfront metastatic or recurrent with 1 prior adjuvant line, but they can't have had first line chemo. And that always confused me until Brian sat me down for about 3 hours and explained it to me. And this is exciting because this has a bispecific with TDXD and versus Pembro. So we'll see if that's actually the second generation of PD-1s is better than Pembro with TDXD. And versus better than our GY018. And then SAC-TMT in, I think, Rob, you said you have this open, Trophus 33? Yes. And Brian, I think, has this open? Yeah.
You're doing a great job.
Thank you.
I just want one clarification because it takes me forever. It's confusing for me. And if it's confusing for me, it's probably less confusing for you. The two differences in these trials, the question in the first line is going to be, do we need chemotherapy? So the Merck trial,
Merck trial.
3, that's up here, is looking at SAC-TMT in the maintenance setting. Everyone's getting GY018, and then they're saying the maintenance setting can make a difference. If you go back a slide, this is looking to see if you could replace chemotherapy in the front line. So, fundamentally very, very different, because if you could replace chemo, then your second line, imagine this, if arm B wins, you're moving careful, we're not even sure if we can do that yet. So it's not only answering the questions that need to be answered in these trials, but does it have to be maintenance? And then treatment would be great. If it's maintenance, how long is maintenance? That's what Rob, the whole thing about ctDNA. You know, we don't know how long we get patients on for IO. How long are they going to stay on ADC? It's a true maintenance setting. So a lot of questions here. Yeah, there's the last thing I was going to say. In the second line endometrial setting, there's 7 things going on. There's an FDA approval for TDXD, HER2/3+, right? So trastuzumab deruxtecan. There's 2 completed trials of TROP2 ADCs. One has a press release that's positive. That's a SAC-TMT led by Keta. The other one is the sacituzumab govitecan led by Ramez. And then there's 4 trials in play that are open accruing. And to Scott's point, how are they going to accrue if one of these others gets an FDA approval? That's the HER2 by BioNTech. There's the B7H4 by GSK. There's the Genmab asset, and then there's the Blue Star asset B7H4 that I think John and I have open. So if the Merck data is positive, it's going to screw up the whole second line portfolio, and then we'll move everything to the first line. But remember how relevant lenvatinib was until Ruby and all those other were positive, and lenvatinib sort of went away as a second line drug. So I suspect ADCs will be in the first line. Sorry for interrupting.
No, that's it's so exciting, and like I think I'm excited for this talk next year at this meeting because it's going to look completely different.
Think you invite yourself to give it again?
Yeah, absolutely. We talked about this a little bit. We're the 3 of us are all real world GYN oncologists, not in ivory tower of academia. What do you see are the biggest barriers to adoption of novel therapeutics?
It's pretty hard to get them approved for our patients if they're not FDA approved. That's hard even when they're on the NCCN guidelines. You have to really fight, which takes a lot of work. I know, Marty, you don't do chemo in your office, but it's tough in the real world sometimes to get what you know is the best treatment for the patients, the amount of fighting you have to do. But it can be done. Unfortunately, we don't have 27 hours in the day. But if you hire the right people to help you out, and actually pharma has stepped up and they will help you a lot of times create the letters that you need to send to the insurance companies and stuff like that, but you still have to take the time to do it, so that's a lot.
They just had the SGO toolkit, the practice efficiency toolkit that they talked about at SGO this year. That has a form letter for approvals. We haven't used it yet, but
I didn't go to that talk, so
yeah,
I'll take a look at it.
yeah, but it's on Connected
Ed.
Connect Ed, yeah.
And we've got about, I'd say, 50 to 70 medical oncologists in our Jacksonville region, and then going out to Gainesville, up to Georgia and down. And we took the approach of let's educate them, and that's one of the reasons why we have our Women's Health Symposium meeting in the fall, to say let's bring them all in and let's bring a lot of this information to them. And we'll still make I'll make the recommendation of, hey, Merv-Bev or this trial that we're going to now open, hopefully with Juju Partners, where we're now a site. But it's a challenge to do. But that's what the approach we've taken is let's bring them in and bring them all together, and let's start to have these discussions.
Anybody having formulary issues with any of these? I know that was an issue initially with dostarlimab and pembrolizumab when the 2 trials came out in 2023. Has anybody experienced formulary issues at some of the bigger institutions? We don't, because we've got 340B pricing and the best P&T committee in Florida, I think. But has anybody run into that? You have?
It took a while to get things off formulary just because we're a smaller institution. It took time to show data and convince the pharmacists. I'm in Tallahassee. We have Capital Health Plan, which is a local insurance, and it takes a while for them to sort of get on board with things. So for a while, I was trying to say, yes, this is a new thing that you need to accept. So
That's been for like every drug?
Most of them are the last ones to get on board.
Wow.
Right.
Yes.
That's right.
Got bought out by FSU, so that might change. But I think the capital kind of world.
In our practice, pharmacist is part of our clinic. So when the patient pharmacist is there, the new regimen comes in. We, for example, our Rosella trial. So we just ask her, hey, this is the data. Just build up a protocol.
That's awesome.
And she finds a way how to get things settled and work closely with our ATPs. I think the fact that we have ATPs and pharmacists help us make a decision, move the needle along. So team build up, let's swing the lane approach that everyone to make shared decision making for patient benefit and utilizing different resources. Partnering with pharmacists is critical for our success. Getting approval is another issue, but at least from a fundamental perspective, that part we were able to figure it out. Now we have a low lag period between initiation of new treatment.
Yeah.
No, I'll agree with everything you said about some of the difficulties, things like that. The only thing I'll add that I've been able to do sometimes, in like the case that we send it, we get rejected flat out, no problem. I've actually gone through the patient assistance programs, and they're giving it away.
Yeah.
Like I'm like, you're giving this patient free drug? I mean, so all the insurance companies are saying no, but we're able still to get it. So I wouldn't stop the fight.
Yeah.
Getting the insurance rejected.
We actually get a lot of free drug. It's actually pretty amazing. However, sometimes when the patients make too much money, there is that cutoff where the farm companies won't support that patient. So in private practice, we probably have a different population than you do from a standpoint. We have way more patients that make a lot of money versus the Medicaid and stuff like that. So sometimes that's not helpful either.
He's not at Broward Health anymore. He's a
My classic patient now is a woman from South America because they can't get these drugs down there.
Yeah.
I had one yesterday from Columbia. Call up, say, "Oh, can you get Merv that don't have insurance? Yeah, how much is it?" If they have the Miami address, they'll get it for free. So they'll come up, they'll get it for free. They'll have, yeah.
I'll tell my patients to get Miami address.
Miami's still in the U.S. I think.
It's very close. Yeah. Thank you again to GSK for sponsoring this. I think it was a great discussion, everybody. Thanks for contributing. And again, be on the lookout. Jenna sent us all those CME links yesterday for everything. So be on the lookout for another email from her. Thank you so much to the GOG for all of these industry-sponsored symposia. I think it's been great having a little bit of extra academic and science during this meeting. And we'll get set up for Marty's talk.